I think we'll get this session underway. So we're in molecular imaging 4 or 3, I can't remember actually, applications. Okay, our next speaker is Professor Hyewon Yoon, who's a clinical associate professor in the Cancer Imaging Centre at Seoul National University Hospital. And the title of her talk is Molecular Imaging Probe Developments, Reporter Genes. Thank you.
Okay, thank you for the kind introduction. I'm deeply thankful to the organizers to have opportunity for me to present our studies regarding the reporter genes. And okay, today I'd like to talk about the reporter gene, basically the nuclear medicine related reporter genes today. And let's start with the briefly introduction of the reporter gene imaging. Actually, the reporter gene imaging is the strategy for the non-invasive assessments of the endogenous biological processes in a living subject by visualizing the reporter gene expression. As you know very well, the actually reporter gene has to be constructed with the promoter and the genes for the reporter. And if you want to make a general construct, just promoter and the reporter. If you want to make a dual reporter or one therapeutic gene or the reporter, then you can make a fusion gene approach or the bicistronic approach. And also, you can use the administration approach and do a promoter. Anyway, in this case, you have to use the promoter. Generally, people use the 2 types of promoter. One is constitutive promoter, and the other is inducible or conditional. When you use the constitutive promoter, it can identify the site, extent, and the duration of vector delivery and the tissue transduction, and also the distribution or the trafficking and the protein-protein interaction. And when you use the inducible or conditional promoter, you can identify the endogenous transcription factor reaction or the signaling pathway, and also the conditional protein-protein interaction. And actually, for the reporter gene for nuclear medicine, it can be divided into 3 different groups. One is enzymatic type, and the other is a receptor type, and the third one is the transporter type. And the first one is the famous the enzyme type of herpes simplex virus, thymidine kinase, and second one is the dopamine 2 receptor, and the third one is human sodium iodide importer. And actually, in our group, the sodium iodide importer is the most focused one. And besides other reporter gene, when we use the reporter gene for nuclear medicine, we also need the probe, radionuclide probe. So each case, we have to have some kind of the radionuclides for the visualization. And also, when we apply to the patient, when we label with the therapeutic gene, then we can remove the kind of exogenous reporters. So that is another advantage of the nuclear reporters. Actually, I will introduce a couple of examples that we did before. This is the most basic reporter gene type. Actually, it consists of P53 responsible promoter and the reporter complex. And we use these reporters. And the drug adrenomycin was reported to accumulate P53. In this case, increase the dose of adrenomycin, and the P53 is increased, and then it binds to the promoter, and the RIS reporter is accumulated and expressed in the cells. And as you see here, when we add the adrenomycin, the P53 amount is increased, and increased amount of P53 can bind to the promoter and produce lots of NIS. So when we introduce this to the cells, and based on the cell number before and then after adrenomycin treatment, we found that the more reporter is accumulated and the iodine signal or the technetium scintigraphy signal is increased after we treated the adrenomycin. And also, the second example is the two dual reporter system conjugated with the constitute promoter. As we mentioned before, constitute promoter can visualize the number of cells or localization trafficking, and that kind of thing we can do with this promoter. And in this case, we used nuclear medicine reporter and the optical reporter, so we can visualize with dual modality. So in this case, we measured the tumor size and the signal intensity from the scintigraphy, and also we measured the tumor size and the optical density. There is a very good relationship between the tumor size and the signal density intensity, especially for the nuclear imaging. The scintigraphy shows the R-square is 0.86. Compared to the optical imaging, because of the penetration depth, I think, in this case, the nuclear medicine imaging, it shows a better relationships with the tumor size. And the third one is the inducible, the promoter that we use with the dual reporters. In this case, we use the retinoic acid responsible element. Actually, this RARE is located in this sodium iodine importer promoter. So generally, for the thyroid patient therapy, to activate the endogenous NIS, people use the retinoic acid for in that purpose. And actually, we transduce this construct to the cells, and tumor cells is xenografted to the nude mouse, and we visualize with scintigraphy and bioluminescence imaging. Based on their numbers, we can see clearly between the small number of cells and larger number of tumor cells in the scintigraphy and bioluminescence imaging. So I briefly introduced this kind of a reporter gene application. And for the application to the clinic, I think it is most frequently tried thing is to take the imaging some kind of gene therapy combined with gene therapy or the adaptive transfusion to the patients. So in this case, recently the chimeric antigen transduced lymphocyte treatment is very well introduced, and the trafficking, the targeting, the activation of that T lymphocyte is really important. So generally, this kind of protocol should require the GMP facility, because the adaptive transduce of the cells or lymphocytes from the patient should be required, and then isolate the cell from the patient. That T cells has to be transduced those kind of therapeutic genes with the reporters, and then expansion and selection is performed, and then back to the patient and where it is, and the therapeutic efficacy has to be evaluated. So recently, Sam Gambier groups reported that they used HSV-TK for this kind of research, and actually they used the constitute promoter and the hygromycin construct in the reporter vector to selection, and also glucocorticoid receptor resistance locus is included because they used GBM patient, and most of the GBM patient treated by the high dose of steroids routinely. So in order to survive the CTL, cytotoxic T lymphocyte, they placed this glucocorticoid receptor resistance locus, and also they placed the interleukin-13 chimeric peptide receptor because they tried to target the gliomas. So this reporter and the therapeutic gene is integrated into transduced into these T cells, and these T cells can recognize the glioma, and also in this T cell express the HSV-TK, and they can use FHBG to visualize the PET imaging. And here is the result, and they showed two patients. And actually, this patient has a frontal parental glioma, and before and after the CTL treatment, they can visualize clearly with the FHBG PET and MR. In the FHBG PET, they showed, after treatment of CTL, they showed clearly in the CTL localization. And also, they showed one patient with the recurrent glioma, and also this patient has a surgical removal before. And also they injected CTL in that recurrent place, and also they treated with interleukin-2 for the survival of the cytotoxic lymphoma. And they also, interestingly, they found that the recurrent glioma, they found the recurrent CTL in the recurrent glioma site. It was injected at this location, but also they found that their surgical resection site, they found the CTLs in that area too. This means that this CTL can go to the pre-surgical resection site. So from this, they conclude that this kind of HSV-TK reporter can easily visualize in the patient with the CTL treatment. So for this kind of application, anyway, when you use the reporter gene, there is a potential limitation because we have to use the expression gene delivery vectors. So that means that there is a transcription activity. Sometimes there is a weak promoter, especially for the conditional or inducible promoter. So in this case, we cannot see clearly. And also, when we express the gene has to express to the modification after the transcription. Post-transcriptional modification is really important. Sometimes mRNA processing and stability is also important. And translation activity is also important. And sometimes the codons, I will explain later, but the gene itself is not optimized for the translation sometimes. So we have to consider about that. And also after the translation, the post-translation modification is really important for the functioning, because our nuclear medicine reporters, most of them are enzymatic and also transporter type. And also sometimes it can work as a kind of 3D structures is important for their function, and also glycosylation, acetylation, methylation, or the ubiquitination and the smolation. Those kind of things is really important for their functions, so we have to consider that. And also, even though there's very successful result from the HSV-TK, anyway, it's from the foreign species, so it can make some kind of immune responses. This kind of thing is potential limitations. So in order to improve this, people try to make weak promoter to improve reporter activity, to add enhancer, activator, or the engineering vector. Until now, there are several kinds of activation is reported, and two-step transcription amplification is really successful. And actually, it contains with the GALFO DNA binding domain and the PP-trans activation domain, and that it generates a lot of PP16TA. And then when we use the weak promoter in front of that, we can add the GALFO DNA binding domains, GALFO binding domains, and then from this strong activator produce lots of persistin, and then it binds to this, and it can amplify the weak promoter. This kind of thing is molecular biological things, and I will skip this. And actually, post-transcription modification, we can engineer the vectors and the treatment to improve fast mRNA processing or just increasing stability. So in our group, we focused on the improving reporter activity for this gene. So let's talk about this. I talked about the codon bias. In order to understand this, we have to understand the concept of codon bias. Codon bias refers to differences in the frequency of occurrence of synonymous codons in coding DNA by mutation or selection. Actually, in the DNA, 3 nucleotide encodes one amino acid, and there is a starting codon and a stop codon, but there is a synonymous codon who it can encode the same amino acid. But the tRNA population is quite different between the species. So if we can change the codon to the frequent existence in the tRNA population in a certain species, then the elongation of translation is really can be facilitated. So in that kind of principle, codon optimization is frequently performed. So codon optimization refers to optimization of translating process by reflecting the tRNA abundances for target species, so it can increase the translation. So actually, there are so many factors to increase the translation and to make lots of reporters, proteins. And actually, this kind of thing we have to consider, but there are so many things. But you don't have to worry about that. There is very good programs that it can include those kind of options. So you can select what kind of thing you have to think, because there is some kind of the conflict between each factors. So you have to consider about that. Tissue content is also important, or the copy number, tRNA gene copy number is important. So when we think about these things, you can select in the program. So there's a lot of good program like gene optimizers. So using this, we optimize luciferase first. Actually, there's so many luciferase commercialized, like Promega cell, PGL 2, 3, 4, that kind of thing is an optimized one for the eukaryotic expression. In order to make a larger commercialized market, they think about they focus on just a eukaryote. We try to make the transgenic mouse, so we optimize this for the mouse, and then we transduced these cells, this construct to the human breast cancer cells and the mouse cancer cells. And in this case, when you see in this figure, we introduced this mouse optimized one to the mouse breast cancer cell line 41 cells. We can clearly see the higher intensity of luciferase signal compared to the human optimized one. Using this, we try to visualize the immune cell trafficking in the nude mouse. Actually, we isolated the splenocyte from the reporter transgenic mouse to the syngeneic nude mouse and see syngeneic mouse and see the biodistribution of the immune cells. And these immune cells are localized to the target organ clearly, to the lung, liver, intestine, and spleen. Most of our, the important thing is that the lymph nodes in each mesenteric and ileum lymph node, those kind of things we can see very clearly in this mouse. Actually, the localization at this moment, the wild type or the LUC2, LUC3 type of the luciferase can be visualized more than 10,000 cells in one locus. But in our experiment, in this case, we can see at least 100 to 10 or 1,000 cells in one locus. We can see, in the case, we can see more sensitive localization in this case. And also, in order to use this, we take this to the skin graft, and then we show, we make 3 allograft and 1 self-grade, and we isolated T cells, CD4 T cells and CD8 T cells, and we see their kinetics. And in this case, CD4 cells last longer, but it also contribute to the allo or the syngeneic graft. But CD8 T cell mostly, the biodistribute in the allo responses. So we inspired this. We decided to make the codon optimization for the NIS gene. And actually, I mentioned before, this gene should have the same amino acid sequence, and then we can change the tRNA population and the other things to be considered. And we changed the nucleotide. For example, in this case, the arginine, we can change wild type, human has a cysteine, but optimized one has the guanine. This kind of changes occurred. And then we generate the synthetic genes. And the codon optimization induction is the maximum is 1, and we generated 0.97 for this case to optimize. After optimization, the iodine uptake is 2 to 3-fold, depends on the cell line increased. And also we transduced this to the thyroid cancer cells, which did not express the sodium iodide importer, and we compared the wild-type NIS and optimized NIS, and small amount of iodine dose we can detect with this optimized HNIS. And we also xenografted this to the mouse and take a SPECT, and we found that this optimized NIS reporter is more sensitive than the wild-type NIS. And also, this sodium iodide importer can be used for the therapeutic purpose. When it treated with iodine 131, we can, after 3 weeks later, we can have a better therapeutic efficacy in this case. This is a SPECT picture, SPECT image with those optimized NIS and the wild-type NIS. So we can see clearly in this figure too. And also, this kind of a reporter gene can be used for the therapeutic purpose, and then oncolytic viral therapy has suggested. Preclinically, adenovirus, HSV, and measles, and vaccinia is tried, but clinically, the most clinical trial is performed with adenovirus and the measles. And recently, the measles virus clinical trial phase 1 is performed in the Mayo Clinic. And so I think if we can use this kind of optimized, they used the wild type ones, but actually we tried to use our optimized NIS for this kind of a therapeutic purpose. And this kind of imaging application, another thing is protein fold and glycosylation, those kind of post-transcription modification. We focus on this, how to use that, and how to improve their reporter activity. So we thought that actually this is membrane protein, and membrane integration is really important for their function. So there is several kind of treatment to improve its integration into the membrane. One is that their folding is really important. So heat shock many heat shock protein can contribute to the folding of the protein. So we tested with the heat shock. First, we checked with the TD tomato fluorescence protein to the NIS protein to visualize easily using the confocal microscopy in the actually in the membrane. We can see clearly in the NIS protein. Actually, we process the image. Generally, we can get this kind of image, but we take a picture with a G-stack and then remove the cytosolic component and then see clearly with the membrane component with the metamorph program. And also we tested with the iodine uptake and the fluorescence intensity with the number of cells, and we can make a very good linear correlation between the iodine uptake and fluorescence intensity in the membrane. And we tested with the heat shock protein expression and the immunoprecipitation. We found that the heat shock protein 70 and 90 and also 40 is related to the NIS folding. And also, actually, the heat shock can improve the protein translation. So we inhibit the de novo protein synthesis, and actually the heat shock can move this protein from cytosol to the membrane to increase their activity. So from this figure, we can see clearly when we add the heat shock, we can see more, membrane NIS can be visualized in this case. And also, we can implant these cells to the mouse and see what's going on. And also after this side, B and D side, we treated with the heat shock. In the case, we can clearly increase this activity. So in the case, we can think about combination with the hypothermia in the NIS application. Maybe it can work. And also, we tested that this glycosylation is really important to the integration into the membrane. And we tested with the two different kind of drug. One is inhibition of glycosylation. The other is the stimulation of glycosylation. So we treated these two drugs, and then we monitored the integration of the NIS protein into the membrane. Actually, we divided the protein from the cells into two portions, cytosolic and membrane portion. And when we treated the inhibitor and the activator of glycosylation, we can see clearly the higher amount of NIS is produced. And in membrane portion, the glycosylation activator treatment can increase the glycosylated NIS. Actually, this size is this, but this upper one shows glycosylated one. So we treated those drugs and then compared the iodine uptake. As we expected, the glycosylation activator treated one shows a higher iodine uptake, and from the fluorescence imaging, we can see clearly the activation helps to integration of the NIS in the membrane. And also, we also stopped the de novo protein synthesis and see what happened, because the cyclic AMP can produce the protein synthesis. So at least we have to know about that. The cyclic AMP itself can improve the cytosol, the translocation of NIS from cytosol to the membrane, and we found that is working. So this kind of limitation has to be, overcome, and this kind of a basic molecular biology stuff is one thing, and the other thing is the immune response. So it'll be better to use the reporter genes from the human, and human thymidine kinase or deoxycysteine kinase, this kind of thing, non-epinephrine dopamine receptor. This kind of thing can be isolated from the human, so we have to focus on this kind of reporter for the clinical trials. So as a summary, I'd like to mention again about the improving reported activity. In order to improve reported activity, first thing is reporter vector engineering, or the treatment for amplifying promoter activity or fast processing or increased stability. And also the improving its function. After this kind of thing is successfully performed, then you can have a better reporter performance. And also, in the future, we have to focus on the human origin reporter and develop for the human isolated one. This is our members and collaborators. Especially, I'd like to express my deepest gratitude to Dr. Jungi Chang. He introduced me the wonderful nuclear reporter gene, sodium iodide importer, and he inspired me lots of experiments. Thank you for listening.
Thank you, Yuan, for a very nice presentation. I think we have just time for one question. Does anybody have a question for Yuan? Please.
Thank you for a nice talk. This is not my field, so I may be way off base, but you're trying to improve the efficiency of your reporting. Is the quantitation at risk? In other words, all the things that you're proposing to improve reporter efficiency, are they all going to improve efficiency equally everywhere, in a tumor cell, in a different type of cell, in one animal or another? It seems to me, I'm just wondering if the relationship, is it possible that the relationship between the reporter and the translation of the gene, the therapeutic gene, let's say, is not going to be consistent? Doesn't that have to be preserved?
Yeah, basically we have to transduce our genes to certain target. For example, when you see a lymphocyte in this case, generally, take the lymphocyte from the patient, and then in the ex vivo, we have to transduce our reporter or the therapeutic genes, and then put that into the patient. After that, only the response of the enhancing the reporter is the cells. It contains reporter or therapeutic genes. So in that case, the contrast can be increased in the target cell only.
I see. Okay. I need to get more instruction.
All right, let's thank you one more time. All right, so, and the final speaker in this session is Michelle James, who's Associate Professor in Radiology and Neurology in the Molecular Imaging Program at Stanford, and it's a great pleasure to introduce Michelle to give her talk, which is titled PET Imaging of Neuroinflammation, Visualizing Neuroimmune Interactions from Mouse to Man. Thanks, Michelle.
Okay, thanks Steve for that introduction, and thank you also to the organising committee for inviting me. So today I'm just going to be speaking about imaging neuroimmune interactions in the context of different brain diseases. And if you had the good fortune to be at the excellent talks by Professor Anna Wu on immunopet or Professor Michael Cassio on developing CNS traces for neuroinflammation, my talk fits somewhere in between those with somewhat of a different yet complementary focus, I hope. All right, so I'm going to start by giving a very brief introduction to what neuroinflammation is and why there's been such an explosion in interest around trying to image this highly complex and dynamic process. And then I'll move into talking about the various PET imaging targets and also the different traces that groups around the world have been trying to make so we can image different aspects of neuroinflammation. And I'll end hopefully with just sharing some new data from my own group where we've been imaging the functional status of myeloid cells, and actually through imaging a biomarker that is upregulated when myeloid cells are in more of a maladaptive state. So what is neuroinflammation? Well, it is any immune process or inflammatory process that is occurring specifically in the central nervous system, in the brain and spinal cord, depending on the disease. It typically is involving extensive microglial activation, and which the microglia are the brain's resident immune cell that are typically the first cells to jump into action whenever there's some kind of injurious stimulation. And there's also astrocytes are playing a big role, another type of glial cell. And also, depending on the disease, there could be some extensive recruitment of peripheral immune cells, both from the adaptive and the innate immune system, and extensive release of different cytokines and chemokines. So typically in its acute form, neuroinflammation can be quite beneficial. It's there for a reason. And it's there to help mitigate pathogens, to clear away damaged tissue. It's involved in healing, so we like it, it's good. But it becomes quite problematic when it's not resolved, when it's hanging around for an extended period of time, when there's extensive release of pro-inflammatory cytokines, there's tissue damage, cell death, and it's a disaster. And this is actually one of the key pathological features that's underlying many neurological diseases. For example, Alzheimer's, multiple sclerosis, Parkinson's, stroke, you name it, neuroinflammation is there. So that we all agree on. Something that people have been debating for some time is where exactly does neuroinflammation fit into the context of these diseases in their initiation and progression? Is it more of a cause or a consequence? And what's interesting is that over the years there's been quite a lot of developments and advances in high throughput genotyping and sequencing technologies, and we now have at our fingertips an abundance of transcriptomic and GWAS data, especially in the Alzheimer's world. There's been a lot of new data that's come out showing actually some surprising results that have shocked the field actually, that in fact a lot of the genes that have come out of these studies are involved in microglial or immune dysfunction. These are the main genes that are increasing a person's risk of developing a neurodegenerative disease like Alzheimer's. And so now all the pharma companies have cottoned on, and they are developing these neuroimmunomodulatory therapies. And so this is important for us as an imaging community, because we have a responsibility to develop very specific imaging strategies so we can visualize these different immune cells in the context of CNS diseases, and not only just their presence and activation, but their functional phenotypes. And that's what I'm very interested in. So let's get into some of the targets that people have been looking at. There are a lot. I don't have time to go through all of them. But just to start, we have the brain's innate cells, so your microglia and your astrocytes. I've spoken a little bit about microglia. Here are a list of some of the targets that people have been looking at. I'll spend a little bit of time talking about TSPO, P2X7, P2Y12, and RAGE. There are so many more, so I encourage you, if you're interested, to read some reviews or come and talk to me. Astrocytes, the main target that people have been investigating is really this monoamine oxidase B, so I'll talk a little bit about that. And then when it gets to the peripheral immune cells, there are a number of targets that are expressed on your peripheral myeloid lineage cells, so your monocytes, your macrophages, your neutrophils. You can see that TSPO is popping up on a few of these. That's because it's not terribly specific for just microglia. That's also the case for many of these. So I want to caution everybody that it depends on the question you're asking when you're choosing your imaging biomarker, and just to really consider that. And before making a range of PET traces, maybe think about the specificity of these certain biomarkers, and especially if you're developing a new biomarker. In terms of the adaptive immune cells, Anna Wu did a great job of explaining the various ways we can image and probe T cells, and also CD20 imaging for B cells. Sam Gambier's group published a really nice paper a couple of weeks back now in JCI, where they developed a new imaging agent for visualizing OX40, which is a T cell activation marker. And that was specifically to visualize response to an intratumoral vaccine. So I encourage you to read that one. And there's also ways that we can image cytokine signaling. So there are imaging agents for TNF-alpha, for CX3CR1. I don't have time to talk too much about that today. There's also a lot of interest in being able to image various aspects of oxidative stress. So lots of different ways for looking at reactive oxygen species and XCT, which is a cysteine glutamate antiporter. There's an imaging agent for that, FSPG. It's been used to image a lot of cancer. It's also really an interesting imaging agent for looking at neuroinflammation and oxidative stress. So I'll jump in now to just some of the TSPO traces. I won't spend too long because Michael already went over a lot of these. But I do want to mention that there are a lot of TSPO traces, and TSPO is a translocator protein. It is by far the most widely evaluated or studied imaging biomarker for looking at neuroinflammation. It does have its problems. I'll talk about those in a little bit. But there are a number of different classes of molecules. PK11195, it was the first generation traces. They're still being used, although they have a lot of limitations with brain permeability and a lot of non-specific binding to plasma proteins. The DPA molecules I worked on with Michael at Sydney Uni, and the PBR28, they were developed by the group at the NIH, Bob Innes and Victor Pike. I'll talk a little bit now just about how these agents are being used to visualize glial activation in Alzheimer's. And I say glial because really TSPO is on both microglia and astrocytes, and it depends on the disease as to how much expression is on each of those cell types. Something to keep in mind. But here we're showing a direct head-to-head comparison from this Yokokuru paper. So comparing PK11195 with DPA713. And you can see quite nicely, I mean, if I was told to judge these images and I was asked to tell who had Alzheimer's and not, I know which images I'd rather look at. The PK11195, it's pretty tough to see a difference there. Whereas with DPA, we're able to pick up on glial activation quite nicely. And then PBR28 has been extensively characterized also in Alzheimer's dementia, led really by Chuck Kreisel, who's over at Columbia now. And he's done some beautiful work, really. He's seen nice correlations between the changes in PBR28 binding and cognitive decline with this. But the problem with these traces, especially the second generation, is that they're sensitive to this polymorphism that occurs in TSPO, meaning that some patients will go in, get scanned, and there'll be no signal because the polymorphism affects the ability of these traces to bind to the receptor. Those people are called low affinity binders or labs. And so we have to exclude those people, which is a bit of a bummer. You can't quantify what you can't see. So people have to come in and get a genetic test before they get imaged. So the group at the NIH actually made this new compound, which is insensitive, well, at least in vitro, was insensitive to the polymorphism. When they went in vivo, however, and this is actually an analog of PK11195 because that is insensitive, but anyway, when they went into vivo, they saw that actually there were some differences between the labs, the MABs and the HABs, so low, mixed and high affinity binders. But the good news is that the binding potential was high enough in the low affinity binders that they don't have to exclude those people anymore. They can get a nice signal, it's displaceable, it's specific. The binding potential in general is still lower than DPA, although DPA doesn't have a stable VT value, probably because of radiometabolites, but we can correct for that. So this is just a little overview of those traces. We've been really interested in understanding whether we can use TSPO PET for predicting cognitive trajectories in stroke and other diseases. Here, I'm going to show you some images from a mouse model of stroke and also our patient data that we've just been collecting. So we use this DMCAO model, which is this distal middle cerebral artery occlusion model. Why we use this, it forms a very specific and reproducible stroke within the somatosensory cortex. And we go, after we perform the surgery, we do MRI. We were comparing two different TSPO traces just to see which would give us the most sensitive and reliable information on glial activation. So we looked at DPA713 versus G180 in both acute and chronic time points. And here you can see we have some results from the 6-day time point and the 28-day time point. You can see very clearly where the stroke is. What surprised me is at 28 days, which is quite a chronic time point, we can still really clearly see where that neuroinflammation is occurring. What I'm not showing you here is that we did a lot of immuno, and we correlated the level of TSPO expression and microglial activation with the actual signal from each tracer. And it correlated very nicely with DPA, but actually not as well with G180. It was kind of a flat line. So in this model, at least, it seemed that DPA was giving us more of an accurate depiction of microglial activation. So we took that into patients at Stanford, and we've just been collaborating with Marion Buckwalter's lab. She does a lot of work on post-stroke dementia, and her thoughts around that is that potentially there is this immune response that's occurring over time in some patients that leads to post-stroke dementia. So we wanted to see, can we image stroke patients a long time after stroke? These patients, it's 8 years after their stroke, so a very long time. And we're starting to see some interesting findings with increased sort of diffuse glial activation in patients that have post-stroke dementia. So just to wrap up on TSPO, while I don't want to throw the baby out with the bathwater, it's still useful. It has problems. It's certainly enabled us a way to really study the temporal spatial dynamics of glial activation in living subjects, which is fantastic. It's just kicked everything off. But it has its limitations, not only the polymorphism issue, but I think the bigger problem is really its limited sensitivity due to this small dynamic range. It only really, in the clinical setting in patients, will go up about 10, 15, maybe 20% if you're lucky. And if you want to be able to image, subtle changes in neuroinflammation, then you're going to need something with a bigger dynamic range for accuracy. It's also not solely expressed on microglia. So if you have questions around microglia or the myeloid lineage cells, then you might need something more specific. It's also on endothelial cells, astrocytes. It's also on cancer cells. And it doesn't really provide functional information on these microglia. And some people might disagree with me, but happy to talk about that later. So other targets that people have been looking at for imaging activated microglia and neuroinflammation, one of them is RAGE, or this receptor for advanced glycation end products. And this is some work, really nice work out of Peter Scott's lab. And his group generated the first small molecule to allow us to visualize this target, which is really involved in facilitating interactions with oxidative stress-related signal transduction molecules like NF-kappa B. And it's also responsible for mediating microglial activation when it's in the presence of amyloid beta, for instance. So interesting target. People have wanted to go after it as a therapeutic target, also as an imaging target. He has shown here, just through some preliminary PET imaging studies in rats and in non-human primates, that has good brain penetration, and also there's binding to regions of the brain that normally express RAGE. I like the name of this tracer, RAGER. And the problem, though, for this particular molecule is that there's high non-specific binding that was found for the autoradiography. And this is using brain tissue from Alzheimer's patients and healthy controls. And that could be to do with the lipophilicity. I believe that Peter's group is generating a second generation of these tracers. Probably the hottest target after TSPO has been, for imaging microglia, has been P2X7, this purinergic receptor ATP-gated ion channel. And there's been a number of tracers for this target. Michael talked about them, so I won't go into too much detail. But suffice it to say that these are probably the top two molecules people are looking at. This GSK molecule has been evaluated in LPS mouse models, also in EAE. It's been translated now to human studies. We're awaiting the data for that. And the J&J molecule shown at the bottom has also undergone extensive validation. Here's some pictures of a non-human primate study showing, I think these are summed from 10 to 30 minutes. In A, it's just a baseline study. B, it's after blocking, so very specific. And this molecule has been translated into people now. We're also awaiting the results for imaging in neurodegenerative diseases. So I'll be fascinated to see that. One of the things I do worry about with P2X7, though, is its specificity. People seem to gloss over it a bit. but it's definitely expressed also in astrocytes, in neurons to some extent, in lymphocytes to some extent. So I think there needs to be more of a characterization in the context that you want to look at in terms of its expression levels and what does that imaging signal really mean. Something that I'm encouraged by is that some people are now developing different PET tracers for P2Y12, which could in fact be a specific biomarker for microglia when they're in more of an anti-inflammatory functional state. So it seems to be a marker of this beneficial inflammation. So there are low levels of this receptor in Alzheimer's, and then when there is some sort of beneficial immune response, it's assumed that it's going up. So that's nice. But another caution here is that it is expressed to a large extent on platelets. So again, just understanding what your questions are and what your imaging signal means. So very quickly, just want to touch on imaging monoamine oxidase B. So people have mainly used the deprenyl molecules, and that's been the majority of the clinical data has been centered around that. So this is a marker of astrogliosis. And you can see in this image here, healthy control, not a lot of uptake. And then in your PIB negative, not a significant amount, but in your PIB positive MCI patients, quite a substantial increase in the binding bilaterally in the frontal and parietal cortices. So that is indicating that astrogliosis is playing an early role in Alzheimer's disease development and definitely warrants the further investigation of imaging astrogliosis. And so I like that Neil Vastev's group have generated this other compound on the far right here. They made this because there's been some problems with the deprenyl molecules, that they're irreversible binders, so that complicates PET quantitation. Neil's group have made the first reversible binder, and that has been translated to the clinic in collaboration with the Orsay group in France, and we should be seeing some data coming out from that pretty soon. So just to wrap up this part of the talk, I've been interested in imaging B cells in the context of neurological diseases. Most people have been looking in lymphoma, but we also know that B cells play a really big role in diseases like multiple sclerosis. And you may have heard about the success of rituximab for treating a certain proportion of MS patients. That's because some people have more of a B cell driven disease, some people are more T cell driven disease. And we'd like to understand who those people are. So we radiolabeled rituximab and we're looking at an MS model, which is EAE. And you can see from these results, naive is an untouched mouse, EAE, you can see down the spinal cord, just dotted red uptake, and also in the brain, which was impressive to me, given that in the spinal cord itself, there aren't a lot of B cells. You can see them dotted around the outside here. In the brain, interestingly, in this mouse model, this is actually a humanized mouse model, by the way, with expressing human CD20. We can see these B cell follicle-like structures like what you see in the clinical setting for MS. It's really nice. And so we correlated our imaging findings with the immuno. We've also started to do some CD19 imaging because we want to be able to image a broader spectrum of B cells in MS in particular. And this is just some of our initial results, showing a nice spleen. And when you pump up the signal, you can definitely see some beautiful lymph nodes. We've confirmed that with imaging and radiolabeling and isotype control and in knockout mice. But just to wrap up in the last 5 to 10 minutes, some of our new data is really, again, centered around MS, and this is something that's very near and dear to me. A good friend of mine was diagnosed, unfortunately, with MS, and it affects more women than men, usually women in their childbearing years. And although there are some really nice disease-modifying therapies available, actually 15 approved now, which is great, the problem is that the guidance for using these therapies is limited. So the neurologist will say, here are all of your options, which one do you want to use? And you're like, well, which one should I use? And they're just going to try it. And then the standard of care is to then try it and then get an MRI a year later. And see if you have an increase in any relapses or if you have new lesions, then maybe they'll up the dose or change it. But really, to me, that just seems like you're playing dice with your brain, and it's scary. And I think imaging could play a role here because we have drugs. It's not like Alzheimer's where we have no drugs. We have drugs here, and we could help to get information about the immune status for these people. It's a really heterogeneous disease too. Everyone's immune signature looks very different. The blood and CSF markers don't reflect exactly what's happening in the CNS. And current PET traces that we have for imaging inflammation, which I've already talked about, lack specificity and can't necessarily tell the difference between beneficial and toxic immune responses. So I think there is this unmet need and something that imaging can really help out with potentially with trying to visualize the relative contributions of different specific immune cells in this disease. And I think this can help us not only with image guiding the therapy, but also in just understanding more about the spatial temporal dynamics of these cells in this disease. So my lab, in collaboration with Katja Andreassen's lab, have identified this biomarker TREM1, which is the triggering receptor expressed on myeloid cells type 1. And it seems to be only upregulated when there is a maladaptive or pro-inflammatory immune response occurring. It goes down when there is more of a beneficial response, when there's healing. And it's only expressed on your myeloid cells, your macrophages, neutrophils, monocytes, which is great. It's linked to both acute and chronic inflammatory disorders. And what's interesting is when you attenuate TREM1 signaling, either pharmacologically or genetically, you definitely see a resolution of inflammatory disorders. So that's been shown in IBD and sepsis, and we've now shown it in MS models. So our aims were very simple. First of all, just validate TREM1 as a biomarker. Just look at what is it actually doing in MS, and then make a prototypical PET tracer. And there are no small molecules for this target, so we really had our work cut out for us. So we have a small molecule discovery program, but in parallel, I thought, well, we'll just radiolabel an antibody and see what we see. So we induced EAE, which is where you give this myelin-specific autoantigen, and then you wait, and sadly, the mice become paralyzed. And so we score them the disease severity based on the level of paralysis. And then we did flow cytometry and PET imaging. So here's the flow results. We know that there are a lot of lymphoid cells in the CNS of EAE mice. We wanted to make sure TREM1 wasn't expressed on those. It's not. Then, surprisingly to me, it's also not expressed on microglia. And actually, we set out to find a new microglia marker, but that's not what we did. We were surprised, and we found a specific peripheral myeloid cell marker, and it's really specifically expressed on those cells. So it's nice that we might be able to discriminate between your central myeloid lineage cells and your peripheral cells. So we went about and labeled an antibody, very specific antibody, and here are some of the first imaging results. So this is just a naive mouse, and it looks like blood flow. So yeah, you're really just seeing heart, descending aorta, just what you would imagine you would see if there's nowhere for it to bind. Then we look at pre-EAE. This is without any symptoms. And you already start seeing, so this mouse will go on to become paralyzed. We start really seeing the spleen lighting up, the lumbar spinal cord, the bone marrow lighting up. This region up here is where we induce the disease. We give them an emulsion with complete Freud's adjuvant, so it really aggravates the skin area, so there's inflammation there. And then we look at low score EAE, and this is where I got really excited. We're in the lab really late at night, and everybody literally screamed when they saw this. But just very clear uptake in the spinal cord, in the spleen, bone marrow. I couldn't imagine a cleaner image. And then in our high EAE mice, so low EAE is where they have tail paralysis, and high EAE is where they have full hind limb paralysis. And again, we just see this beautiful signal that's just popping out at you. And you think, well, is that really specific? Well, we did a TREM1 knockout mouse, and what we did here, what was interesting, was that most of the TREM1 knockout mice didn't get sick, so it seems like it could be a therapeutic target. But some of them did, and we had some at score 1 and some at score 3, but this is what they look like. They just look like the naives, where there is inflammation there, and these mice actually do get better. It's just blood flow. So I'm pretty happy about that. We then go on to do specific image analysis where we can segment out the spinal cord and use a 3D mouse brain atlas. And I won't go through this, but all of this quantitation reflects what we saw in the images. Highly significant differences with percent ID per grams, like 20% in the spinal cord, which is really nice. And then you might think, well, Michelle, is that just because there's increased blood flow? And is that really, like, are we really seeing that? Well, we remove the organs, we perfuse the mice, we remove all the blood, and we remove all the organs, and we see still these significant differences in the lumbar and thoracic spinal cord and in the brain, also in some of the peripheral tissues. And then we do ex vivo autoradiography, and you can see again, like, I mean, I have to draw a line around the naive. There's no TREM1 expressing cells in the CNS normally. And then it's just blowing up in the low, and then in a knockout mouse, it's very low. So this is my second last slide. We also did a quick study just to see how does TREM1 PET compare to TSPO PET in the same cohort of mice. And what we saw was this. I mean, I can let you guys be the judge. This is what a TSPO PET signal looks like. And why is there so much going on here? Well, it used to be called the peripheral benzodiazepine receptor. So there's a lot of this receptor in the periphery. And it obscures a lot of what's going on in the spinal cord and in the spleen. It's hard to see the spleen. But then we take we quantify it, and then we'll take the organs out as well. It doesn't seem like there are significant differences in the spinal cord. There is in the brain, which is cool. But at a similar level to what we were seeing with TREM1, maybe a little lower. Then if we compare directly the ex vivo gamma counting of the tissues, so looking at ratio of EAE to control, the fold differences for TSPO are around 2. But for TREM1, they're greater than 2 in brain and greater than 10 in the spinal cord. So that, to me, was really exciting. But I think we could be imaging very different things here too. I think TSPO is more on the microglial or glial activation, whereas this is more the peripheral infiltrating cells. So in summary, on this project, TREM1 looks like it could be a nice, highly specific biomarker of these myeloid-driven immune responses, this innate immune activation. We've looked at it in a number of models now. And TREM1 PET seems to be quite promising as well for visualizing this toxic immune response. And we have some ongoing work in this arena, looking at therapy monitoring, translating this imaging agent. But I hope that I've been able to share with you a little bit of background about what neuroinflammation is, why it's important that we image it, why there's so much increased interest in this area, and some of the targets that people have been looking at and those that are emerging, and the importance of really, as a field, us all working together to really identify these biomarkers that will tell us about not only whether the cells are present and if they're activated or not, but what are they actually doing. And I think we're still a ways to go from getting there, but I think we're making some nice progress. And I'll just quickly thank my lab, in particular Dr. Aisling Cheney, who's done a lot of the work on TREM1, and Emily Johnson, and all of my collaborators and funding. And thank you. Thank you.
I guess my question is, because typically antibodies don't cross the blood-brain barrier.
Right.
So are they, it looks like they would be picking up the antibody in the periphery, in the spleen, and what you're looking at is trafficking into the,
Exactly.
so that's...
Yeah, because we have to wait. At the 2-hour time point, we don't really see that kind of imaging data, and we wait it's a day later that we really start seeing it.
Or do you see the spleen right away in a couple of hours, and then later on the signal comes up in the spinal cord?
You're 100% right. Yeah. So we're getting a free ride on the cells.
Awesome talk, thank you.
Thanks.
I'm just curious, in your stroke work in rodents, do you have any idea why there's such a difference between DPA and G180?
It's a good question. I don't know. I don't know why. They're two different. They're structurally unrelated. Usually, I've done quite a bit with G180 in rodents, and it's looked great. It's actually developed for rodent imaging. So I just don't know.
Okay.
Yeah.
So is that, you've just seen these differences in the stroke work? Have you seen it in any of the EAE work? Or?
I haven't seen it in the EAE, and I haven't properly validated it in the EAE yet. We're going to just do a bunch of staining and flow data. But in Alzheimer's, we've done a lot of characterization, and it worked beautifully. It correlated with microglial activation. It wasn't correlating with GFAP. So, yeah.
Is it comparable to DPA?
In Alzheimer's?
Yeah.
Yeah, it is.
Yeah.
It's still still DPA is a tiny bit better, which surprised me because it's C11. So I thought that it wouldn't be, but the signal to background just seems to be better. So the lipophilicity of G180 is higher. So maybe it's just that there is more non-specific binding, and you get a bit more background. That's my guess.
Okay, thank you.
Yeah.